Good morning, everyone. My name is Yifan Yang, and I'm currently a PhD candidate in Penn State University. Although I'm unable to be physically present due to a visa issue, I'm really grateful to OHBM for providing me with this opportunity to share with you some insight from my recent research, which is about memory encoding in the form of the replay and its potential predetermined nature. Intelligence relies on the fundamental process of learning and memory, and the hippocampus plays a very critical role in these brain functions. A well-studied phenomenon is the memory for places where the hippocampal place cells were thought to be the key neural elements. During the spatial navigation in the environment, these cells shows the ability to form spatially constrained firing field or spatial receptive field to encode the animal's current position. On the screen, you can see an example of a place cell with the place fields that located at the early position on a linear track. The activity of multiple place cells together thus constructs the neural representation of the experience when the animal was running through a maze. And during resting, these place cells recap past a sequence but in a temporarily compressed form called replay. The typical duration of a replay event varies between 100 to 400 milliseconds. Such replay events often coincide with the hippocampal sharp wave ripples, and it has been proposed as a mechanism necessary for memory consolidation. The nature of this hippocampal firing sequence is a matter of active research. Increasing evidence suggests that these sequences are not solely induced by external experiences, but rather emerge as a result of internal brain circuits. An intriguing mouse study found that with the mouse running on a stationary running wheel, the CA1 neurons consistently fire at different times, showing the time-specific firing patterns, so-called time cells. Remarkably, these time cells display the firing sequence despite there is no change in the mouse's physical position. This really suggests an internal nature of the hippocampal firing sequence. Another compelling evidence comes from the discovery of preplay. It has been found that the temporal firing sequence of place cells, which are typically observed during a novel spatial experience, are actually present during the resting period, even before the experience. This phenomenon suggests the hippocampal sequence is not only internally generated, but its temporal order is also predetermined. Therefore, comparing to traditional view that the hippocampal sequence are solely driven by external environmental cues, Buzaki and Linus propose an alternative theory. That is, this sequence could be internally generated and coordinated by some hypothetical ordinal sequences. Then, an intriguing question is, what could be these internal ordinal sequences? So, initiated by this thought, we explore one potential self-generated neural event that may shape firing sequence of place cells. Recent studies showed that the resting brain exhibits brain-wide infraslow activity in a highly organized spatial temporal pattern. In the fMRI, they are shown as the waves propagating along the cortical hierarchy gradient. At the meantime, in the large-scale single neuron recordings, we found that it manifests as highly structured neural event that involves majority of recorded neurons across different brain regions. Because of its sequential nature, this neural event we call the neural spiking cascade. It is self-generated, highly coupled with arousal state measured by the pupil size. Most importantly, it is strongly correlated with the ripple event, with the highest probability occurring around the center of the sequential cascade. Therefore, it may suggest a potential relationship between the cascade and the memory functions, specifically in the context of replay events, since both of them share common sequential natures. So, to explore these possibilities, we conduct our analysis using the Allen Visual Coding Mice dataset. There are 2 nature movie sessions included in the task design with a 30-minute spontaneous session in between. Each movie session contains 30 repeats of a 30-second nature movie. We found that the hippocampal CA1 neurons exhibit time-selective response with specific time field in the movie. And these time-selective neurons collectively form the sequential firing patterns during the movie watching. And during 30 minutes spontaneous stationary period, we derive a template matching algorithm to detect replay of previously identified time-selective neurons. The replays are present in 2 directions, with the forward replay colored in red and the reverse replay colored in blue. The averaged replay pattern are shown in panel C, which closely resemble the actual sequence experienced during movie, as shown in B, but in a much temporal compressed form. We also did a statistic test for the number of replays that can be detected, and found the CA1 replays significantly more than other control cases, such as replay in the visual regions. Consistent with the literature, hippocampal ripple events are more likely to coincide with the detected replays. So, what could be the potential link between the replay event and the sequential cascade? We found that for lots of time, the detected reverse replay occur at the sharp transition point of the cascade, which marked as the yellow triangle in this example. This key observation is general and supported by the distribution of replays across all cascade cycles, as shown in panel B. We found that the reverse replay tended to align more closely with the cascade centers, while the forward replay exhibits a different modulation, spreading around the cascade. This modulation pattern was found to be replicable across different mouse, suggesting a consistent relationship between the replay event and the temporal dynamics of the cascade. Despite that, the hippocampal ripples are indeed more prominent at the center of the cascade, they can also occur at the other faces as well, as suggested in this example. Then, when we use a higher temporal resolution and zoom in to examine the fine-scale structure within the different phases of the cascade, it's clear to see the presence of global events occurring at a much faster timescale, coinciding with hippocampal ripple events. These micro-events are bidirectional, with one direction aligning with the original cascade, termed the M2P microcascade, representing the direction from the negative delay neurons to positive delay neurons. The opposite direction was referred to the P2M microcascade. Importantly, we found that the reverse replay were found to co-occur with the N2P microcascade, while the forward replay were associated with the P2M microcascade. This finding suggests a critical role for the bidirectional microcascade in facilitating the temporal organization of the hippocampal replay events. So in summary, our study revealed 3 key findings. Firstly, the CA1 neurons shows time-specific response during the movie watching. These time-selective neurons collectively fire in sequence, similar to those place cells or time cells. Secondly, this firing sequence can be observed in a temporally compressed form replayed during the resting period. And finally, we found that this replay actually coincides with brain-wide spiking cascades. Specifically, the forward and reverse replay were observed embedded within 2 types of microcascade with opposite directions. And altogether, these findings highlighted the self-generative nature of the replays and the hippocampal sequence in general. and suggests that their temporal structure is governed by the ordinal sequence within the resting state circuit dynamics, manifest as the spiking cascade. Okay, thank you all for your attention. I have provided my email address here, so please don't hesitate to reach out if you have any questions. Once again, thank you for your time.